GATE 2022 Life Sciences, Section S Microbiology, question number 71. Phenolics acts as disinfectant by dash. Rupturing plasma membrane followed by leakage of cellular content. B. Bond formation between adjacent pyrimidine bases. C. Forming adduct with amino acid and unsaturated fatty acid. D. Alkylation of proteins. Okay, this is the NPTEL online course reference for this question. So these are the few definitions you need to understand before. By antisepsis is defined by any agent to a living tissue, okay, for the purpose of preventing infections, okay. So any agent, antisepsis is any agent which we use. It could be either chemical, okay, it is a chemical agent which can be used to a living tissue for the purpose of preventing infection, okay. Like we can use Dettol, or we use different antiseptics, creams, okay. Then we have decontamination. So decontamination is either to destroy or to reduce the activity. Remember this activity of microorganism, that this is decontamination. Then we have disinfection. So disinfection or disinfectants are the chemicals or any physical treatment that destroys the vegetative microbes, viruses, but they cannot destroy spores or in any inanimate surfaces. So remember, disinfectants are those chemical or physical treatments, okay. So for chemical we have disinfectants, we have phenol, ethanol, and they could be physical treatment also. So we have various physical treatments, like we can treat at 120 degrees Celsius, right. So when we autoclave in microbiology, the temperature reaches to 120 degrees Celsius at 5 atm psi, 5 atmospheric pressure at 5 psi, okay. So these are the physical treatment for disinfecting the microbes or viruses, but they are not able to, these treatments, these disinfectants are those which are not able to destroy the spores. Then we have sanitation. So sanitation is just reduction of the microbial load, okay. So the sanitizers which we use, they'll just reduce the microbial load on the surface, okay. It can be either surface or, okay, it is the surface or the inanimate surface to a level considered accepted for the public health purposes, okay. So sanitization generally occurs in hospitals, in the areas. So during COVID, we used to have sanitization, okay, whenever there was a patient seen in a building or any locality. Then we have sterilization, okay. This is very common term used, sterilization. So it is a process intended to kill or remove all the types of microorganisms, and which includes spores, viruses, okay, and the probability of surviving them is very low, okay. So sterilization is, we are trying to eliminate or kill all the microorganisms, including the spores. In disinfection we have only destroying of vegetative microbes and viruses, okay. In sterilization we completely destroy the spores, okay, and there is very low probability of surviving any spores or microorganisms. And then we have pasteurization, okay. So pasteurization generally kills the non-sporulating microorganisms by hot water or steam. So pasteurization we do it for various as we know, during milk is pasteurized. So the main idea for pasteurization is to ensure the quality of milk, and by this ensurement I mean that the nutrition value is is still upholded, okay, by these means of killing. So this is generally done by hot water or steam. So these are the mode of action. This is very important. These are the different modes of action of disinfectants. So let's start here with alcohol, phenols, and these are the different chemical agents. We have alcohols, formaldehydes, aldehydes. We have phenols. Okay, what they do, they disrupt the cytoplasmic enzymes. Okay, they disrupt the cytoplasmic enzymes. So alcohol, phenol, they have highly reactive OH group. Okay, they have OH group which is able to dissolve many of the enzymes which are hydrophobic. Okay, they can dissolve hydrophobic enzyme, and they can so essentially all this phenols, alcohols, and formaldehyde, they can dissolve various hydrophobic molecules, and one of the major hydrophobic molecules we know is the cell membrane, right? So the phenols are capable of dissolving, so as you can see, we have aldehyde phenols. These are capable of dissolving the membranes, right? These are capable of dissolving the membranes as well as the hydrophobic enzymes which are present in the cytoplasm. Okay, this is very important. So phenols, as you can see, these are also class of phenols. They can dissolve hydrophobic molecules. This is very important. Phenols are the alcohols which will help to dissolve the hydrophobic molecules. And then we have aldehydes and chlorines, which mainly targets the mRNA. Then we have different peroxides, hydrogen, formaldehyde, and aldehyde, which are able to also form adducts or form reactive ROS, reactive oxygen species, which target the DNA and they can lead to breaks, DNA breaks. Okay. And also hydrogen peroxide, as you can see here, it is capable of targeting the ribosomes. Okay. So when we speak of phenols, the main targets are the plasma membranes. Okay, because plasma membranes are hydrophobic in nature, right? They are made up of fatty acids, and thus their phenolics can act as a disinfectant by rupturing the plasma membrane, and after the leakage of the cellular content, it can further target different enzymes. It can target enzymes and other hydrophobic molecules in the cytoplasm. Okay, so the correct answer is A, rupturing the plasma membrane. So all the phenols, always remember phenols, ethanol which we use, phenol, ethanol, formaldehyde, these are all present in the sanitizer, disinfectants which we use. Okay, these generally rupture the plasma membrane, okay, of the microorganisms. Bond formation between adjacent pyrimidine bases. You know the example, the thymine dimers. So if this is a thymine, adjacent thymine in a DNA, they can form a dimer, right? They can form a dimer like this, and this thymine dimer is usually formed by UV light, right? This we studied in the last lecture. UV light is able to form between adjacent pyrimidine bases. Thymine is a pyrimidine, right? So so this is thymine dimers. Thymine dimers are formed by UV lights. Okay. Forming adduct with amino acid and unsaturated fatty acids. So there are different chemical reagents which forms adducts. Okay. And alkylation of proteins. There are also different disinfectants which alkylate proteins, but not phenol. Okay. Phenol reactive group is just OH. Okay, always remember, see the structure OH. So it won't be able to form any adduct with the amino acid or alkylate the protein. It doesn't have any alkyl groups to alkylate, and the adduct formation is also not possible, right? So the correct answer is A, rupturing plasma membrane followed by leakage of the cellular content.